Welcome everyone to the workshop on copy number analysis in Bioconductor. And I'm just going to very briefly introduce the three people from my lab who are going to be giving this workshop. And it's a great example of a variety of expertise coming together and developing some really good materials and workflows. So Marcel Ramos is a software developer and also member of the Bioconductor core team. He has been doing a lot of work on core infrastructure, including the ragged experiment data class, which can be used for copy number and mutations. Ludwig Geistlinger, who has just finished another workshop as a postdoc, who has, in addition to his work on enrichment analysis, has done work on population copy number analysis, including things like what you think of as GISTIC doing, that is identifying regions of common overlap and doing eQTLs and such. And finally, Sehyun Oh is also a postdoc and has done some very significant copy number analysis on the application side for absolute copy, allele-specific copy number analysis of tumors in the Cancer Genome Atlas and from targeted sequencing panels. So with that, I will go ahead and hand it off to you.
Great. Thanks a lot, Levi, for this introduction. Yeah, just like maybe to get things set up well in the very beginning, there was also just this one comment in the BioC Slack channel. It's quite confusing with all these different windows here I have to navigate through. So the main point that I would recommend to go to is really here this website that I posted here in the chat that will give you the fully rendered version of the workshop with all the contents that Marcel, Sehyun, and myself will be speaking about. And then maybe after the conference, if you want to try out things a little bit in more detail, you could use the GitHub version. And for like direct hands-on, you can run here this instance given username and password here in the chat. With that, I'm actually going here to this fully rendered vignette and say, of course, that I'm very happy to like give here the second edition of this copy number variation workshop on this year's conference. It's a great workshop because we are doing it together with Marcel and Sehyun, dear colleagues from our lab. And we have basically three parts here. We have a part which Marcel starts off, which basically introduces a general class for those of you that are familiar with summarized experiment, which has similarity with summarized experiment, the ragged experiment, for generally storing genomic location data such as CNV data. I will then take over and introduce our new CNV Ranger package that basically takes a ragged experiment and allows to do some more downstream analysis, such as integration with gene expression and quantitative phenotypes. And then eventually Sehyun will talk a little bit about a more advanced application example of copy number variation analysis, introducing the PureCN package, which allows allele-specific copy number variation analysis, which we do a lot in genomic cancer analysis to estimate different tumor characteristics such as tumor purity and ploidy, which is possible to indeed infer from copy number variation data. So splitting it into these three parts, I just wanted to point out when talking about copy number variation data, we are basically talking about genomic region data. That means in the most simple case, we have like genomic regions given a chromosome, a start point and an end point. So these are genomic intervals. And of course, this is a ragged scheme here because we might have a different set of genomic regions for different samples. And I refer to these kind of regions by saying CNV calls. A little bit of literature that is relevant for this workshop, the CNV Ranger material that uses a ragged experiment, that is this bioinformatics paper from this year. Sorry for the noise in the background. We have these paper here from Sehyun, which is relevant for the third point, where it comes to inferring tumor characteristics from CNV data estimated from whole exome sequencing data. and more advanced, where we make use of these cancer genomics application of CNVs. Just impress here in cancer research is this paper where we applied all these analyses for a real world data analysis in the context of subtype of ovarian cancer. All right, with this, I think I hand it over to Marcel, who started us off with a ragged experiment.
Thanks, Ludwig. I will share my screen if I may.
Yes, I stop share.
Okay, you should be able to see my screen now. So I wanted to introduce to you the ragged experiment class, and we'll start here with a small live demo of how to generate your own ragged experiment class. This class was, I guess, born out of the need to represent a set of ragged ranges as a matrix-like format. So usually when you have working with range data, you represent this data using genomic ranges or G-ranges list. So in order to take that structure and convert it to a matrix-like format, we use ragged experiment. So the first thing we're going to do here is to load the package. And I've already spun up my instance, and hopefully you've been able to open that on your own side of things. So I'll do library ragged experiment, and then we'll go in with our G-ranges example here. So I'm creating a G-ranges using the G-ranges constructor, and hopefully we've looked at this already. And the G-ranges constructor takes a vector here where you have the genomic range. So in this example, I'm using chromosome 1 from 1 to 10, and the strand can be, in this example, it can be anything. So here I have my G-ranges function, open parentheses. In the middle, I have a vector, a named vector. So the first element in that named vector is A, and I will go into my second range here, CHR1, 8 to 14. And this, you can add any number you like for the example. This is just to show you how you would construct a ragged experiment from scratch. So that is our first range. You can also have metadata associated with these ranges. For example, in our small example, I'll have a score. So, and maybe Ludwig can comment more about what a score could be in a G-ranges here. So, and then we have another metadata column, say for example, germline and somatic. So this could be mutations in the mutation annotations in this G-ranges. So that's our first sample. So, and for sample 2, we will go into the same thing, G-ranges, and we'll start with D equals, let's see here, so chromosome 1, 11 to 14, and E equals chromosome 1. So we're going through just to show you how a typical set. So you have 2 G-ranges, and so we'll look at what sample 1 outputs. So this is the G-ranges show method, and sample 2 is similar. Here I could have used one range or 2 ranges. I'm going to add my scores as well for these. So, and the type. So
Just just because I have been prompted to say something to the score that could be, of course, like, for example, your copy number variation state, with maybe 2 being the normal diplet, 3 being a gain, 1 being a 1 copy deletion. But as Marcel also talks here about mutations, it could be a quality score or a B allele frequency or something like this.
Thanks, Ludwig. So we can also have our call data annotations. So these could be, this can be a data frame if you load it from the S4 vectors function or package. The data frame can also have annotations. So yeah, so I'll go to my script because I think I'm running a little bit short on time, but essentially the first thing you do is you obtain a G ranges, and other ways that you can obtain a G ranges is through a data frame type of tabular read-in. And you can use these helper functions from genomic ranges, such as make G ranges from data frame or make G ranges lists from data frame if you're importing your data in a tabular format. And then once you have those G ranges, you can either create a G ranges list or simply enter those as part of the constructor function call here. So you may also have additional information regarding the samples or participants. So you might have participant IDs and whether the participants control or case, and those can also be part of the ragged experiment object here. So there's pretty much a lot of flexibility here for representing different types of data, as Ludwig said, mutation data included. So, and you can find this script here in the inst scripts folder once you are in the instance. So you can go to the inst scripts and you'll find the, and you'll be able to rerun the thing yourself as well. So.
Great, thanks Marcel. I maybe go over with sharing my screen. So I think once that we have a regular experiment, once we have this information there, I have here a little figure that maybe also illustrates a little bit further what Marcel was just doing, that given very general genomic location data here, such as chromosome start and end position for a different set of samples where I have additional characteristics in the call data, as Marcel just explained. So I might have maybe, if I'm talking about cattle, I might have weight of the cattle and also their feed conversion ratio. So how does the nutrition that they take in convert into muscle mass? What we would typically do is to take such a simple format where I have the sample identifier here and maybe like a copy number state, with 1 being a single copy loss and 0 being a homozygous deletion, so maybe a 2 copy loss. Then I would import this into R and store this in dedicated bioconductor structures. And as Marcel said, first option is like a list-like representation where I have for each sample here, for sample 1, these 2 calls, and for sample 2, I have here these 2 calls. So I have here a list representation with each element of the list being a sample study. And the other thing that Marcel introduced, if I maybe would like to have a rectangular matrix-like representation for an access that is not a list-like access, but rather a matrix-like access, then I would wrap a ragged experiment around it, where then would have here this column by rows, rows by column representation, seeing here, okay, these first 2 regions in sample 1, I have here a single copy loss, 2 copy loss, these other 2 regions are not covered in sample 2 in this sparse beginning representation in a ragged experiment. But the good thing is about the ragged experiment it allows you here to apply a number of summarization function to basically condense this sparse representation. And making use of the specific functionality that these bioconductor structures such as G-ranges list and ragged experiment allow us, I can actually do quite a number of downstream analysis, which is here available in this CNV Ranger package, CNV Ranger at Bioconductor. And the main functionality that is here is that on the one hand side, often I want to summarize like these individual calls across my population. So I have maybe like hundreds of samples, cancer patients, and I basically want to, you know, define regions of copy number variation by summarizing these individual calls. And there are different approaches for that in the literature. One thing, this is the so-called CNV Ruler approach, basically just trims a region based on density. It basically checks, okay, what's the coverage here in the region, and are there like outliers that maybe here are not covered overall. So the density is maybe less than 10% of my samples analyzed, and then it would basically just clip off here this part and report this as a summarized region. Another approach taken is to require sufficient reciprocal overlap between the calls. And that would maybe result in a more fine-grained summarization. I would maybe say, okay, these two calls here have enough mutual overlap to be summarized within one region, and then I would maybe say, okay, these two guys have enough mutual overlap to be summarized into one region. And then in cancer, where I often have a lot of, you know, like random passenger mutations as opposed to driver mutations, I'm also interested in defining regions that are recurring, so that that exceeds just like a random distribution. When I take these calls and stack them just like by location and magnitude over each other, and then I'm asking, does this magnitude exceeds when I would just randomly place these calls across the genome in order to identify here recurrent region? That is this region, this GISTIC procedure that you might be familiar with when you are in cancer genomics, for example. Another thing that you then want to do once you have specific regions in your genome where you say these are concentrated regions of copy number variation is of course to ask about the functional implications of these copy number variations. And one thing that you would often like to do is to ask, are these regions overlapping with known genomic features, for example, known genes? And there I would again have genes, here are 3 different genes represented by genomic intervals. And I would basically ask to which extent do they overlap with my copy number variations. And of course, a certain overlap would always be expected, right? If I put random regions onto the genome, some regions will just by chance overlap with genes, just because I have genes scattered all over the genome. And again, I could use here a little bit of a significance assessment. So I could basically do a permutation test. I repeatedly draw random regions from the genome that correspond in size and chromosomal location with my CNVs, and ask how often do I actually observe such an overlap when compared to these random distributions. So it allows me to assess, is there here a depletion or an enrichment for overlap between my copy number variation regions and my genomic features such as my genes. And then I, of course, want to do association tests, right? I want to on the one hand side, associate often with gene expression to basically associate, are there genomic structural changes? Are there changes, copy number changes that are indeed detectable on the expression level? Thus, a copy number variation change induces an expression change. And for that, I would then maybe also have for these genes here read counts from an RNA-seq assay. Here I basically have for these 3 genes, for these 2 samples, I have read counts mapped to them. And then I would basically do here a classical differential expression testing between different states in like defined genomic windows across genes. So I would basically distinguish here between different effects that a CNV could have of a gene, most intuitive or dosage effects where CNV coincides with the gene, such a local effect, and you would, for example, check whether an amplification here indeed would result in an increased expression of the gene. But then you have also proximal effects where you maybe have copy number variations in promoter regions or other neighborhood effects, or maybe even distal effects where you have copy number variation in enhancer regions that also affect your gene expression. And you might also want to do classic GWAS testing where you're basically not testing SNPs, but now a CNV with quantitative phenotypes in your call data. So a lot of functionality here in this CNV Ranger package. And I think I'm showing a little bit what can be done here. One thing that you basically want to do is here the summarization. Therefore, you have this population ranges function, which you give a G ranges list or a ragged experiment. So you would basically start with a very simple format. You would read in a file. This is here an example from a publication which is contained in the package, and you have a very simple 5-column format, the chromosomal location, the sample ID, and then the copy number state, just like coded in this form here. You have 0 being a homozygous deletion, 1 being the heterozygous deletion, 2 normal diploid, and then 3 and 4 for corresponding 1 or more copy gains. And then having this data in a G ranges list or a regular experiment, we could start with summarizing these genes and these copy number variation regions. And one thing that we could do is we could basically say, okay, summarize these calls into regions of concentrated copy number variation, clip off regions that have a density less than 10%. It was this example where I said, okay, I require here a certain amount of samples to be falling in these regions. Regions that are not covered by a sufficient number of samples I clip off. And then I would also do such a GISTIC-like recurrence estimation by setting here estimate recurrence equals true. And what it gives you back is basically, again, a G ranges object where it says, okay, in this region, out of 700 samples that I, in this example data, analyze here in total. So I have here 700 steers. This is Brazilian cattle data. And out of these 700 steers, I found 100 samples to indeed show a copy number variation in this region. And most of the time, this has been a gain. It's an argument to the function when it says it allows for a little bit of a deviation, but here it's 10%. So there might be some loss calls in there, but a vast majority of calls have been here a gain. You might also have loss, or you might have where you actually have like heterogeneous copy number variations. It might be a loss in some samples and a gain in others. And then you have basically here this recurrence, whether this exceeds what you would gain by random sampling. And we could visualize this. We could say, okay, take these regions, these recurrent regions, and plot the areas of recurrent regions on chromosome 1. given UCSC genome identifier. Here I take the Bos-Taurus genome assembly version 6, and looking here at chromosome 1, and then it basically shows you, okay, here are your copy number variation regions spread across chromosome 1. We have regions here in blue that are predominantly again, having here the frequencies of samples that carry a copy number variation in this region. We have here in pink loss regions, and then in green where we have both. And on the other hand, we would really see, okay, where we have here this annotation track, we see, okay, these are regions that exceed our random background level, which are recurrent regions. So that is one thing that I can do to really identify regions in the genome that show concentrated copy number variation. So I think it's about my time to pass on to Sehyun, but another thing that I maybe just shortly show, of course, you also want to, once you have summarized these individual CNV calls across a population, to do more things, such as overlap analysis with genes or a CNV expression association analysis. And for that, you also have dedicated functions to, for example, do here something like a CNV oncoprint, taking your CNV calls in your G-ranges list or your regular experiment and your gene coordinates. And then you could basically make here such an illustration which shows which genes tend to be affected by copy number variations in many samples. So we basically see here the samples in the columns and the genes in the rows. And I see, for example, that MAP2 is either experiencing a 1-copy loss in these samples, or sometimes also a 2-copy loss, being overall deleted in either heterozygously or homozygously deleted in up to 40 samples. So that summarizes very nice the overlap between genes and copy number variation regions. And you have more to also then integrate with gene expression data. There is example with TCGA data here, where you basically look at a specific copy number variation region here on chromosome 1, and you basically test for differential expression in the neighborhood. These are here specific genes on the plus or the minus strand, and you basically see here their fold change and the p-value when you test expression based on grouping on the copy number variation state in this region. And you, for example, see here that there are certain genes that indeed show a dosage effect because they are locating directly on this copy number variation regions, but you also see that you have a lot of neighborhood effects here, which is something that has been observed previously by ourselves, but also others. Okay, and with this, I would maybe stop here and let you share your screen, Sehyun.
So till now, actually, we saw how to work on CNV data itself. And there was a really good segue, because one of the questions is whether there is any bioconductor package that can do the CNV de novo discovery. And this is exactly the section for that. So in this last part is about what to do if you want to get the CNV information from the raw data. So I will briefly introduce a CNV SNV analysis workflow using the bioconductor package called PureCN. So I just want to make sure note that the most important feature of this workflow is that, unlike other existing workflows, it doesn't require matched normal sample that's corresponding matching sample to the corresponding to the tumor sample, which can be much more applicable for clinical settings where matched normal is not readily available. And you can go, so the vignette we've been using, it has quite a detail. So due to the time limit, I will go through little slightly simpler version in the PowerPoint format here. So, and before I go through more, you can find any more detailed information you can find in PureScience vignette or PureScience paper. And the paper, my paper, Ludwig introduced earlier, that the workflow describing the benchmarking workflow itself, you can find this in this paper link here. And the part is, the other thing I want to mention is the workflow itself is actually implemented in Terra workspace as Tumor Omni CNV. And here, Terra, where Terra is the cloud-based genomics platform, and which will be actually covered more in the other workshop this Thursday morning. So if you're interested in, then you can join there too. And there is a detailed script is also available GitHub repo. The thing is, like, a Terra workspace has a little more detail, and then also example file format is available there, so it might be a little easier. So just FYI. And so this to find out your own CNV information from your own data. Okay, so this workflow practically requires 3 inputs. So first is bat file to find out the captured region. And the second type of input is a process match normal BAM file. So important part here, process match it. So it doesn't need to be the same, it doesn't need to be a normal from the same patient's like tumor. And then this process match normal is required to figure out the background noise. And then the last part is, of course, your tumor sample that you want to check the CNV information. So these are brief summary what's happening in this step, and I will just go each step very quickly in this workflow overview. So this is practically same, and then it just I put a little bit of like name of the tools, and then the intermediated output files between the different workflow, different steps. And so first you need to get the target information using the BAT file. And then this will generate an interval file from BAT file that you need to use in the downstream analysis. And next you need to generate the pool of normer, which will capture recurrent technical artifacts. And last, you need to detect both germline SNPs and somatic SNPs from the tumor BAM file. For that, we use the MUTECT. And once you have, those 3 are not dependent on each other's, so they don't require any info as from other workflow or other steps output. So you can run those 3 in parallel. And after that, you need to calculate the GC normalized coverage, which will be used to correct the library specific biases. And then you will build a normal database. And once you did all those things, and then you can finally run PureScience, which will normalize, segment, and determine purity and ploidy. So then that output will have the CNV information. And I will say this is not a simple workflow, but good news is at the production level, as long as you're using the same capture kit and the same sequencing protocol, you need to process only tumor BAM files. So all the other bad and then normal BAM file information you can use. So you need to run only this highlighted part if you want to repeat. And then based on the modularity and then dependency on inputs from other steps, this whole workflow is split into 6 workflows and implemented in Terra, as I mentioned. So I will just briefly show you how it looked like. Let me see. Yeah, so if you go to the Terra, so this is the public work. So the terminology is, I think I don't have enough time to explain those terms, but briefly, this is the place, this is the public page, and then there's no control data set. So as far as you have just free account, you can access all this information. So under the dashboard, you see I put all the information about this workflow and then how to prepare the input files. And data part, for this one specific one, to make it public, I use the synthetic data set, but you can bring any of your data set or any of your own data set or any data set Terra is hosting, which is very long list of data. And then the workflow is where I told you there is, actually there is 7 because I have one more downstream analysis part. But you can go to, let's see the part that workflow that create the pool of normer. So you can have the access, can access all these actual script, and then like the runtime environment is in the Docker container format, so you can easily access it or public document, fully documented. So you can actually see the detailed script to run this, and then you can provide your input in a very easy way. So this is how require the input, and then you can assign the data you want to put, and then there is a way to link the output to the input file, like using different prefix. So you don't need to hassle around the different directories or like thinking about creating file name to like link all those data. So this is a workflow, and then I also have some of the Jupyter notebook written in R that's for the 4 of this for upstream pre-processing, and then 1 as a downstream analysis. So if you go here, you can have all those, you can have access to all those things. And so example, the output, so once you do all these things, how it will look like. So you can view you can see so you can see the purity and ploidy plot, and then it just score the most likely purity ploidy combination in this summary plot, and then you can plot it in the histogram format. And then, by the way, this can be readily available in our RStudio server. And then, so that's the like basic purity ploidy information. And then you can create the CSV file with create curation file that you can make a note on your result. And then you can get the amplification and deletion. So core alteration is extracting major copy number alterations to amplification and then deletion. So you can just extract that extract that result with one function. And the last one is like call LOH. So you can detect this function will extract the detailed loss of heterozygosity information by region. So this is the result. So PureCN give a lot of easy access to the data, and actually the whole data is in one RDS file, but you can extract all these things, and there is easy visualization functions too. So this is how you will use and how you will do if you want to get CNV information from your own data. And I think that's it from CNV side.
Great. Thank you very much, Sehyun. I think we have still 15 minutes for questions at least. And I see there are a number of questions here in the poll section. Feel free to enter more questions as of now. Maybe we start with, maybe I share my screen for this purpose shortly. There's one question is that maybe could go to Marcel in order to stay with the questions in the order of the presenters. Could you quickly explain what does these text indicate as such? So given this character representation, what does it stand for, and how to translate this maybe into a genomic range in Bioconductor?
Yeah, so G ranges can be represented in many different ways, and one of these ways is through a string, and you can annotate your string that way using the chromosome, the range, and the strand, and then convert that into AG ranges in our example earlier.
Great. Marcel, would you maybe also like to say a couple of words about the Q-reduce assay function, which I think is super useful when working with ragged experiment?
Yeah, and I can share my screen so I can show the graphic.
Great. Because we still have a little bit of time that we could use for that.
All right. So there are a number of functions that will allow you to sort of reshape the original data that you had as the ragged ranges. And the Q-reduce assay allows you to take a window and say a region of interest and reduce all of your ranges in your data to those, or restrict to your data to that window. So the Q-reduce assay does that. Compact assay tries to figure out which ranges coincide with each other and align them in that particular order. And the sparse is the sort of the default representation where each sample is sort of stacked, as you can see in the image, on one after the other.
Great. Another question which seems to be for me is, if I got this right, this is the CNV Ranger approach. This summarization approach is useful for downstream analysis once you already identified CNVs. Are there bioconductor packages for a CNV de novo discovery? Well, there's a lot of things to say here. I think also PureCN, the package itself that Sehyun introduced, allows to actually do copy number detection itself from whole exome sequencing data. I'm just pointing out, as I said in the beginning, there are many different ways how to identify CNVs, either experimentally based on comparative genomic interpretation. And these days we also infer CNVs a lot from either SNP array data or next generation sequencing data, such as whole genome sequencing, or in Sehyun's case, whole exome sequencing data. There are a number of bioconductor packages, of course, for that purpose. One thing, one very popular old school package, but still used a lot this year, this DNA copy package. You could always find more by going to the corresponding copy number variation BioC view. If you go here on copy number variation, you'll find a lot of packages for this purpose. On the other hand, actually, the CNV Ranger package itself has a dedicated section on that. So if you click here on the vignette of the CNV Ranger package, and you go a little bit and what does the package actually do? As we said, it's a package for downstream analysis, and I give you some recommendation what I think works good. For SNP-based CNV calls, then I would recommend using PennCNV for de novo CNV detection. I tried out myself for sequencing-based CNV calls, CNV Nader, which applies a read depth approach, or Lumpy, which basically combines evidence from split reads and discordant read pairs. There has also been a lot of development in recent years. So you don't like only apply one tool. You might have want to assemble CNVs from different tools. So for that reason, or for that purpose, I would recommend Ensemble CNV or Fuserism. I think this gives you quite a selection from which to use your tools. Another question, and this is maybe for Sehyun. Can you please explain how to interpret the ploidy and purity plot?
So purity and ploidy, that summary plot is like summary plot. I will interpret that like based on the likelihood score. So even though it's not, it's like it look like a heat map, and then in this specific example, specific example, it seems quite obvious, but sometimes like likelihood score, like rank it somewhat not less intuitive, but I will follow that because that likelihood score is coming from a lot of other information under the hood. So I will follow that I will follow the label based on the likelihood score. So I will pick that. So for example, in the vignette, I will say that like this sample is most likely with the purity of 0.6 something, and then the ploidy of roughly 2. So I will just interpret that way. And yeah. And by the way, I had a few other questions on the chat, so I directly answered to that. So if I'm not saying it right now, please check. And then if I miss, let me know. I'm going through it right now. But.
Okay, great. Thank you, Sehyun. I think there is another question that goes maybe to Marcel and myself at the same time. It asks, could the ragged experiment be used for chip-seq, attack-seq peak calls? And this is the first part of the question that maybe I can answer. And then there is the second part of the question that maybe Marcel can answer. What is the advantage of ragged ranges versus all ranges with a presence-absence matrix for peak calls, or in general for genomic regions, for example? So first part of the question, yes, exactly. I mean, the ragged experiment is a very general data structure for storing ragged genomic location data. And this could be SNPs, this could be CNVs, this could be chip-seq calls, this could attack-seq. Also, right now we have a lot of single-cell attack-seq, and their ragged experiment is, of course, also a dedicated structure to store these calls, simply due to the fact that we have here this ragged design. We have different calls, different peaks for different samples, and the ragged experiment basically allows this to represent this efficiently. Now, Marcel, would you like to answer or say something on the second part?
Yeah, one thing I forgot to mention is deciding whether you should use a range summarize experiment or a ragged experiment. So when you have all your ranges measured across all samples, I think the range summarize experiment is the ideal class for that. If you have differing calls per sample, then the ragged experiment would be the appropriate structure to use. I hope that answers the question.
Great. There is another question that maybe is going to me. The question is, how can SNPs be identified that locate within CNVs, if I understand it correctly? And I think for, if I understand the question correctly, and for that, I maybe share my screen here one more time. And I think the main question here, and that is maybe the reason why we use these bioconductor structures, because something like the G ranges universe allows a lot of genomic region algebra between genomic intervals. And one essential functions when dealing with G ranges is the subset by overlaps function. So as I would understand the problem, what I would have on the one hand side would be maybe here these summarized CNV regions as one genomic region object. And then on the other hand, I would just have such a G ranges object which stores my SNP, which stores my SNPs again with a genomic location. And then if I wanted to find these SNPs that overlap or that locate within a CNVR, I would just say subset by overlaps, SNPs, and CNVRs, and that would give me back the subset that locates within CNVs.
Actually, I saw some question to me, I think.
Cool.
So there is a question that is there a limit to the kind of genomic data that can be accessed in Terra app? No, pretty much there is no limit. So right now, especially if you are bring your own data, you can bring any kind of data. Currently, it holds all the different kind of genomics data, including like FASTQ, BAM, VCF, or BAT file, or are available. And then they're, available meaning that for example, TCGA consortium should have that kind of data already. But if so, then it's not a problem to have that, use that in the platform. Also, this different kind of like patient metadata, like age or like tumor or not, and all those things are also available in Terra. And one other thing is how to perform CNV calling and analysis for single sample tumor omnicase. That's absolutely doable. So in my workflow I showed you, there will be just only one tumor. The thing is you need the process match normer. So whatever your tumor is processed, any normal sample that went through the same process. So usually about 10, 20 is enough, 10 normal sample is enough, and to filter the technical bias. The efficiency of 10, 20 is good enough. Around over more than 50, it kind of saturate. The have more normal doesn't really help anymore. So I will say that you need a BAT file, your tumor sample, and then like something like a 10 to 20 of process match normal sample. Then you can just run the same workflow. And there's one more, I think it's related to this CNV analysis too, but most CNV analysis is performed on tumor sample. Are there any recommendations for analysis of non-tumor samples, any factors to be considered? That part, I'm not quite sure about it. And just, so I don't have a clear answer to that. Just one side note is actually, because pure CN doesn't use its matched normer, and then we are assuming that the tumor sample is somewhat contaminated. So the purity of like somewhat mix, like 50% of normal and then tumor mix, actually called with most confidence. So if it's too pure or completely contaminated, then it has a harder time, because underneath it just assumes that there will be some contamination of the normal, and then use that as to like part of it. The tool is using to get the replace that lack of matching normal. So that's just a side note I want to mention, but sorry, I don't have direct answer to that specific question. But that's it.
But that was nevertheless a great answer. Thank you, Sehyun. I see here 2 more questions. I also see that we're coming to the end of the session. So let me maybe take these 2 questions here on CNV Ranger before closing up this session and encouraging more questions also via Slack or the provided emails to either one of us. One question that I see here is that what are my thoughts on Sequenza? I have to admit, I haven't heard of this tool yet, but I will look it up after the workshop and we'll get back to that if I have an impression what the tool does. And I would share my answer, or basically give my answer then here in the chat, or also if you are on Slack in this BioC workshop question channel. On the other hand, there is a question, is it possible to use different reference instead of a USC reference genome? Can I, for example, use a gene code or an ensemble reference? There's a bit to say to this again. Maybe I share my screen for another minute, going to the point where you basically take here gene coordinates, and we have here in Annotation Hub, or you could also use EnsembleDB Bioconductor package to get gene annotations from different genome databases. For example, here I obtain ensemble gene coordinates using the Annotation Hub functionality. So in general, yes, this is possible and you can convert. I think the question was going a little bit more towards how about making these genomic regions plots and which kind of genome identifier? So here to note is that this is based on GWAS functionality, which is another bioconductor package, which is very useful for doing these kind of plots. GWAS bioconductor, so under the hood, this package is called. And as far as I know, I might look this up a little bit further, but I think you have to provide a valid UCSC genome identifier. There might be some ways to provide here an object instead of a character identifier, and that could be a way to make this work. I might look this up and follow up a little bit more on this question as well. All right. With that, I would like to thank you all for the attention, and I would say we stop the meeting here. Further question can be addressed through the Slack channel. All right, thanks everyone.
Thanks.
Bye.